Hey everybody, in this video we are going to talk about the difference between spontaneous mutations and induced mutations. So an induced mutation essentially is any mutation that is caused by an external agent. Now, when we say external agent, it could be a chemical agent, like everyone knows tobacco smoke is supposed to contain many chemical compounds that are carcinogenic. They thought to cause cancer, and they do that because they cause mutations in our DNA. So chemical agent can be one type of agent in the environment that can induce mutations. The agent can also be physical. And usually when we talk about a physical agent, we are talking about radiation. And we'll talk more about these in a moment. So what do we mean by spontaneous? So a spontaneous mutation, essentially, I think the easiest way to understand this is, any mutation that is not induced by an external agent. So essentially anything that's not an induced mutation is a spontaneous mutation. And why do we even bother distinguishing between these two types of mutations in an introductory genetic course? So I feel the reason is it's important for us to know that mutations can cause spontaneously. So, we could be doing everything right, and just the processes by which a cell needs to survive and to live and do whatever it does, sometimes, mistakes can happen and cells can get mutations, and those aren't necessarily triggered by something in the environment. So mutations can be spontaneous and mutations can be induced, and we will take a look at some examples of induced mutations and spontaneous mutations in this video. So one of the first types of spontaneous mutations, well, let's cover spontaneous first. So a type of spontaneous mutation, which is actually important for a range of genetic disorders, is called polymerase slippage. So this would be DNA polymerase during DNA replication. So polymerase slippage, and this can happen in repetitive regions of the genome. So for example, let me take a short repeat of DNA, CGA, and the opposite strand would be GCT. Now, let's pretend this goes on. And here are the two repeats, right? So CGA followed by CGA. And let's say that's 5', 3', 5', 3'. Now imagine that this goes on and on and on. So let's say there are like 30 repeats of these 3 nucleotides. So that would be a repetitive part of the genome. Maybe it's in a gene, maybe it's between genes. Many of the genetic disorders occur when these are within genes. So now what is polymerase slippage? Instead of me diagramming this, let's take a look here at this one. Well, I've already diagrammed it. So, okay, so here we can see I have the GCT strand already drawn, and let's imagine this is being replicated. So this is the strand undergoing replication, and imagine the polymerase is adding nucleotides to this strand right here, the growing strand, and this is the template. I messed up, didn't I? CGA. So no mutations are occurring, and let's say it's continuing down here, and then all of a sudden something happens. The polymerase falls off the template, and as it's falling off, it drags this newly synthesized strand off the template as well, so it detaches it. What caused that polymerase to fall off? I'm not really sure. Maybe a helicase got in the way, or DNA gyrase or something was in the way or bumped into it. Who knows? Who knows what caused it to fall off? But it fell off, and it caused this to come apart from the template. Say the end of the strand right here to come apart from the template. So that might look something like this. Okay, so, you know, the DNA has to finish replicating, and what scientists think is that sometimes, you know, when the polymerase is loaded back on and this is reannealed down here, it can reanneal imperfectly, leading to a hairpin loop within this structure right here. And let me show you what that could look like. So the very end of the molecule anneals to one of the repeated regions, and this can only happen because of the repetitive nature of the strand. So we have this CGA down here, and it should have been up here, but it reannealed down here. And now we get this hairpin loop right here that is not annealed to the template. And structures with a lot of G's and C's tend to form these hairpin loops. And scientists think that is the case because, let's see, do I have a pair here? You can see this pairs with this C, pairs with this G pairs with that C. We have GC, CG here, and these hairpins are thought to be very stable. So genetic disorders usually occur in these repetitive regions, and genes with these repetitive regions with a lot of C's and G's, because scientists think these hairpins, when they have lots of C's and G's, if they form, they tend to be relatively stable because of this intramolecular base pairing or hydrogen bonding here. Now, okay, so what happens? So replication continues, and the polymerase loads back on here, uses the A, or the 3' hydroxyl on this A right here, and extends this strand. So now the cell has to identify this and fix it one way or the other. So maybe it will cut it out and then there won't be a problem. But another possibility is that there's a nick made in here, and then this is used as the template to fill the gap, to fill the resulting gap. And if you look in the notes, you see a pretty nice diagram of what happens when a nick is made in this side. We end up with a gap, and then we have to actually add more nucleotides to this strand down here. The end result is we've added a lot of nucleotides to this sequence. Nucleotides that weren't there, we've added them. And as a result, we've increased the length of the repeat region. We've increased the number of nucleotides in this region. And so polymerase slippage, when it occurs in regions with repeats of 3 nucleotides, lead to these TNRE disorders, or at least are found associated with TNRE disorders. And these are trinucleotide repeat disorders. I should say TNRE stands for trinucleotide repeat expansion disorders. So trinucleotide repeat expansion disorders. And some common ones are Huntington disease. Maybe some of you have heard of these diseases. Fragile X syndrome, in spinocerebellar ataxia, So just some examples, and there are several others. And again, these occur because these trinucleotide repeat regions tend to expand, and we think they expand because of replication slippage. Okay, so that's replication slippage, and that's a form of spontaneous mutation that can occur in cells without any chemical or physical agent causing it. Another common type of spontaneous mutation is a result of deamination of cytosine. So this is just, the nature of cytosine, that the nature of its structure leads to it, often reacting with water to be deaminated. And what does deamination mean? So deamination occurs when a amino group removal of an amino group from a molecule. So, what's an amino group? Okay, well, let's take a look at the structure of cytosine. So the structure of cytosine is going to look like this, if I get it correct here. I don't make any mistakes. And I normally don't diagram the C's, but okay, why not? I feel like I'm missing an N here. Put a C here. Okay, I usually don't put the double bonds in, but I'll do it this time. Let's say this goes down to the sugar, the 5-carbon sugar. Let's put some hydrogens here. And here's the amino group. So a nitrogen with 2 hydrogens there connected to this carbon here. This is the amino group. And cytosine tends to react with water, at a relatively low rate, but common enough to be a problem. And as a result, we get a structure that looks like this. So that amino group is gone, and we're replacing that with an oxygen, double bonded oxygen to that carbon. And I think that should be the only difference. Still attached to the sugar. Okay, so that's not too much of a big deal, right? This is cytosine, and this is this cytosine with an oxygen. No. So this is uracil. And so why is it a problem? Because cytosine, it pairs with guanine. So if cytosine was in a template and DNA was being replicated, a guanine would be placed on the opposite side of cytosine. So guanine would be paired with cytosine. But if the amino group is removed, replaced with an oxygen, well, now this is uracil. Uracil, if it's in DNA, if it's in the template during DNA replication, well, uracil pairs with adenine. So an adenine would be put in across from the uracil. And then eventually the cell will fix that uracil, but what will it put in its place? It will remove uracil and will add thymine. So we've had a mutation as a result of the deamination of cytosine. So again, those are spontaneous mutations. Deamination of cytosine occurs at a low rate in our cells, but often enough to be a problem. Now, we have in our cells things called reactive oxygen species. We're talking about another form of spontaneous mutation here. So reactive oxygen species, these are chemicals like hydroxyl groups and superoxide, hydrogen peroxide. Essentially, they're chemicals with oxygen in them that are also highly reactive, that are highly reactive, meaning they like to interact with other chemicals and change the bonding structure or things like that. So our cells have reactive oxygen species, and they use these chemicals for signaling and other things, but they have to keep these levels at a relatively low level and controlled. If those levels go up, the cell loses control of them, they can damage DNA. And because they're there at a low level, you know, sometimes mistakes can happen, and sometimes these things can damage the DNA with, you know, without levels necessarily increasing. So one of the most important mutations caused by reactive oxygen species in the cell is when guanine is mutated by a reactive oxygen species, also called an ROS, mutated to 8-oxoguanine. So why is this a problem? Well, guanine pairs with cytosine, right? 8-oxoguanine pairs with adenine. So you can imagine that if 8-oxoguanine was in a template and DNA was being replicated, well, adenine would be put across from 8-oxoguanine in the newly synthesized DNA, in the newly synthesized strand. Eventually, when the cell got around to fixing this, it would change it to a T. And that would be a mutation. So we're sensing a theme here, right? When we're messing with the bases, or some chemical is modifying a base in the DNA, if that modified base pairs better than than the original unmodified base does. Okay, I'm screwing up my delivery here, but what is the pattern? Modified base pairs better with, let's say, non-canonical base, the one that normally pairs with the unmodified base. I think that's a good way to say that. Modified base pairs better with a non-canonical base. Then we can lead to mutation, unless that modified base is fixed. I'm calling it modified, but let's say mutated. Mutated base pairs better with a non-canonical base, then we can have a permanent mutation unless the mutated base is fixed before replication. So, okay, and I think in the notes, in the printed notes, we have something about, this is kind of nice. I want to see the study on this. Let me see, 10,000 or something. Something about 10,000 of these types of mutations. One second. Where was this? Oh, I'm glad I checked my notes here because I skipped over something. I got you. Okay, so this is a problem, and we're going to talk about a type of mutation that is thought to occur in each cell tens of thousands of times next. Okay, so that's 8-oxoguanine. Now, the next one, a spontaneous mutation I want to talk about, which is very common, is called depurination. Now, depurination goes hand in hand with pyrimidation. Switch pencils here. Be prepared for the tip to break. So, spontaneous mutations, two other types: depurination and depyrimidation. So for this, understand these, we need to remember what the purines are. We have our mnemonic device, pure as gold. Purines are adenine and guanine. Okay, so depurination means loss of guanine or adenine base. Now this is just the base, the 5-carbon sugar, the phosphate backbone, that's still there. So just loss of those bases. That can occur, and that's called depurination. Depyrimidation would be loss of cytosine or thymine. Now, depyrimidation is less common than depurination. It's because of that bond between the 5-carbon sugar and the nitrogenous base. That bond in purines is a lot less stable than the bond in pyrimidines. So depurination is common, and so loss of, I guess, depurination is thought to occur tens of thousands of times in each of our cells. So, and those have to be repaired before DNA replication, otherwise we can get a mutation. So let me diagram this thing right here. So I'm diagramming one strand of DNA, and this is the phosphate backbone right here. Let's say this is the 5-carbon sugar, those little vertical lines there. And you might speed up, watch the video in double time while I diagram this until I finish this diagram. A, G, A, G, A, G, G, G. I'll put a T in there, why not? T, C, T, T, T. So I'm pairing these up. Double bonds here, triple bonds there. Triple bond, double bond, triple, triple. These are hydrogen bonds, remember? Okay, so depurination would be, okay, we're losing this G. So the G is gone. It falls off because the bond between the 5-carbon sugar and the nitrogenous base is not that stable. And so we're left with a 5-carbon sugar here with no base. Now, why is that a problem? Right? Okay, we can separate these strands right here. And if DNA replication occurs, you know, separate the strands, use each strand as the template, replicate the DNA. When the polymerase gets to this base, there's nothing there. There's nothing in the template. So it can be skipped, and that will be a deletion, or, you know, maybe a random base is put in there, and that will lead to a mutation. So we can see how depurination would be loss of A or G, or depyrimidation, which would be loss of a T or a C. Could lead to a permanent mutation. Again, so if the cell can identify these and fix these, you know, before replication, then there won't be a permanent mutation there. Okay, so that's depurination and depyrimidation. Now let's talk about types of induced mutations. And we have a new term here, DNA adduct. In DNA adducts, this term is kind of confusing. Essentially, anytime a chemical is added to the nucleotide, we call that a DNA adduct. So an example of a DNA adduct would be this compound called ethylmethane. Well, a DNA adduct is caused by a compound ethylmethane sulfonate. So geneticists use this compound as a mutagen because it, you know, causes DNA adducts, and the DNA adducts lead to mutations after replication. So what happens exactly? So ethylmethane sulfonate can be abbreviated EMS. And, you know, I'm not sure where this is found in the environment. Maybe it's found in tobacco smoke too, you know, tobacco. I don't know. But I know geneticists use this as a mutagenic agent for inducing mutations in a model organism, like E. coli or a fungus or even a plant. Possibly even mice. I don't know about that for sure. I don't work with animals. So instead of diagramming or producing a new diagram, I'm going to show you how EMS reacts with guanine nucleotides, or changes guanine or adds a DNA adduct to guanine. So here's the structure of guanine. We have a sugar right here. We have our 4 nitrogens, 1, 2, 3, 4. Okay, so this is what it normally looks like, and this normally pairs with cytosine. Well, EMS, when EMS is present, what happens is we're adding an ethyl group. So CH3 and CH2 attached to this oxygen over here. So we have this oxygen double bonded to cytosine here. Well, over here, we have it single bonded to the cytosine and single bonded to the ethyl group right here. So we have this big molecule hanging off of the guanine now. And as a result, this pairs with adenine. So pairs with adenine instead of cytosine. So again, this is how we get mutations, because EMS adds something to the guanine, causing it to pair better with adenine than it does with cytosine. Did I say better? Well, it definitely pairs, I'm assuming it pairs better, but it definitely pairs better with adenine than it normally does. And it can lead to incorporation of adenine during replication when a ethyl guanine is in the template. Okay, so that's one type of induced mutation. Now, tobacco smoke and other things contain lots of different chemicals that can damage our DNA in similar ways. And those are relatively easy to, conceptually understand. You add anything to the DNA and you just make it more difficult to replicate DNA in a proper way, in the correct way, or incorporate the correct base every time when the DNA has been mutated and it doesn't form the correct hydrogen bonds between the base pairs. So let's also take a look at physical forms of induced mutations. So physical mutation would be caused by ionizing radiation, or a physical source, physical mutagen. So ionizing radiation. So what is that? So we have a couple examples here. Cosmic rays, and maybe if some of you have already taken introductory physics, you have covered these things and you're probably more familiar with them than I am. But some forms of ionizing radiation are cosmic rays, gamma rays, and X-rays. And I believe cosmic rays and gamma rays we're mostly protected from our atmosphere and maybe the magnetic field around Earth. I think that is the case. So, but if we were not protected from these, and if we should come across a source of these, well, they would severely damage our DNA. And X-rays, well, we come across X-rays all the time when we go to a doctor or dentist. Many of us are X-rays multiple times per year, or a couple times per year. So this type of radiation is considered ionizing because the photons, again, I hope you have physics already, so you know what a photon is, but a high energy photons break bonds in molecules. So the photons in this type of radiation have so much energy that they're able to easily break bonds in molecules should they contact those bonds in the molecules. And if the high energy photons break a bond in DNA directly, it's called direct damage. So ionizing radiation can cause direct DNA damage if the high energy photons hit the DNA molecules directly and break bonds in the DNA directly. Or they can cause indirect damage. So what does this mean? Well, this means it hits another molecule, say in the nucleus, and forms what we call a free radical. So breaks bonds in the other molecules in the nucleus, say non-DNA molecules, but breaks bonds in them, creating free radicals. So free radicals, what are those? Those are chemicals with loose electrons. So electrons are supposed to be paired to be stable. And if you have a loose electron, well, that electron's going to look to pair with something. And if it's trying to pair with something in the DNA, it can break a bond in the DNA. So that would be indirect damage. The high energy photons damage another molecule in the nucleus, not the DNA, and that molecule becomes a free radical, and that free radical damages the DNA. So pretty bad stuff right here with ionizing radiation. Non-ionizing radiation can also cause damage. So that's kind of strange, right? Well, this guy is high enough to break bonds. How does non-ionizing radiation, how can that break? I mean, how can that cause DNA damage? Well, UV light can be ionizing and non-ionizing because UV light spans wavelengths of 100 to 400 nanometers. And some molecules, well, they ionize at 120 nanometers, and others ionize at 280. Some don't ionize in any of these ranges here. So UV light can be ionizing or non-ionizing depending on the molecule. But with respect to DNA, UV light is non-ionizing or causes a non-ionizing form of DNA damage, and that is called pyrimidine dimer formation. Pyrimidine dimer formation. So here we're not really breaking the bonds, but the nitrogenous bases in the pyrimidines or in the pyrimidine nucleotides are absorbing the energy and the bonds are being rearranged. And that's why I believe we call this, or geneticists call this, a non-ionizing form of a damage, or damage caused by non-ionizing radiation. We're not really breaking the bonds, we are rearranging the bonds. So pyrimidine dimer, this happens when UV light hits DNA, and there are two T's, two C's, or a C and a T next to each other in the strand of DNA. So not across from each other. These have to be next to each other in the same strand. And because of the atoms within thymine bases, these are more commonly turned into pyrimidine dimers than these guys down here. So this is the biggest problem right here. But we can also get pyrimidine dimers formed by two adjacent C's into C and a T. Okay, so let me talk more about that. Again, not breaking bonds, but rearranging them. And let's take a closer look at what this means. A pyrimidine dimer, what is that? So, let's go with this. I have this nice diagram here. I figure this is quicker. So let's say this up here is a piece of our DNA in a random gene. And here's the sequence of the top strand, sequence of the bottom strand. And we can see we have two T's side by side right here. 5 carbon sugars are here, 2 T's are right here. So a photon of UV light comes in and hits this area, some here, I'm not exactly sure how the physics works here, but causes a rearrangement in the bonds. So now we have 2 covalent bonds formed between these adjacent thymine residues. So these bases here aren't normally connected to each other, right? They're normally hydrogen bonded to the complementary base in the opposite strand. When we get these bonds formed between these adjacent thymines, that causes a structural change in this strand right here. So these can no longer properly hydrogen bond with the adenines on the opposite strand. And keep in mind that, I have the DNA diagram nice and linear like this, but DNA is a double helix, right? These strands are twisting around each other. And when we have these bonds formed between here, the strands don't twist properly. So we have a big structural change in this DNA. So if we were to separate these and this was to be replicated, DNA polymerase, would have a hard time putting A's across from these T's. And as a result, when these are present, they are a significant source of mutation. And we're going to see a disease, xeroderma pigmentosum. And this is the genetic disorder in humans. And if a human has this genetic disorder, they can't be in the sun, because UV light causes DNA damage, and I think they have a 2,000-fold higher incidence of cancer. And we'll talk more about this in a second. It just shows how important it is for our cells to identify these types of mutation caused by UV light before replication occurs. And individuals with this disorder are lacking a key DNA repair process that seeks out and fixes these types of pyrimidine dimers that result from exposure to UV light, which happens when we go out in the sun. Okay, so that's another form of induced mutation, and that is it for our discussion of spontaneous versus induced mutations. Again, all this is written up in the notes, so if you did butcher a little bit of the delivery in this video, take a look through the notes and work on the problems, and send me an email if you were confused on anything. Okay, see you in the next video, where we will talk about different ways to classify mutations in genes. Okay, see you soon.